So I just a quick update on where the Cure CASK campaign is. So it's supported by 3 charities, so CASK Research Foundation in the UK, AECF in France, and ACNRF in Australia and in the US. And although we obviously told the community about it some time ago now, about having UC Davis very keen to look into the CASK gene, and us creating what we call the Cure CASK campaign to try and raise funds, we've obviously been quite quiet, and that's because we've been really busy behind the scenes. We've had 4 peer reviews of the proposal from experts in the arena. They've come back. They've then had to be responded to by the UC Davis team. They then had to go back to our research review committee, who reviewed everything, and then gave their formal recommendation back to the foundations on whether to proceed. So you'll be pleased to know that was all very favourable. So hence the webinar today. So we're all good to go. We just now really need people's support and understanding of what Kyle and his team and Jill, who we can't see, want to try and do. So they're going to try and explain it in as simple terms as possible. Just a few people have recently, and myself actually, seen in the news, it's been in the news quite a lot about gene therapies for rare diseases. So I just wanted to very quickly kind of explain how this is gene therapy, but it's not like the kind that have been hitting the news recently. So the ones that have been hitting the news tend to be called gene replacement therapies, where they're replacing the gene. So just to kind of start it off, this isn't replacing the gene, and Carl will explain that. Sadly, we have investigated gene replacement therapies. Sorry, I'm just letting people in. It's not possible at the moment to do gene replacement therapies on CASK from what we have learned. So we have contacted quite a few different eminent scientists. The CASK gene is large because it is neurological. Currently, there doesn't seem to be the appropriate technologies to enable the kind of therapy that you've been seeing to be successful in the news. So because of that, that's why we're really focusing on this X reactivation, because it's the only gene therapy that we know of that could be possible. And it's all a big if and a big could, but could be possible for CASK, the way that current technology and current understanding goes. So that's why we're supporting Kyle and Jill, and Dave couldn't make it, but he's also part of the broader team. So the one thing about CASK, even though it's a bit of a difficult gene in terms of the therapies that are kind of out there, is that, according to Kyle, it's a very good fit for what they're trying to do. So it's a really good candidate for X reactivation, which is why they're so excited. So anyway, enough from myself. I'm going to pass you over to Kyle. And he's going to introduce himself, and hopefully you'll be able to hear Jill, and they're going to tell you what they intend to do.
Yeah, great. Thank you, Laura. And everybody can hear me okay? And or see me okay? Okay, I'll take the non-response as a yes. And so my name is, as Laura introduced, my name is Kyle Fink. I am a, I see a hand up already. I'm an assistant professor in the Department of Neurology here at UC Davis. My lab is located in what's called the Institute for Regenerative Cures in our, and as part of that is the Stem Cell and Gene Therapy program. Oh, they can't see me. Let's see. My camera is on.
I can see you. I think it might be their issue, not yours.
Okay. Okay.
I can use you, Kyle.
Oh, cool. All right. And so several years ago, what we really started to do here at UC Davis, and in between collaborations between Jill Silverman and Dave Siegel and myself, was really to start to focus on rare genetic disorders and neurodevelopmental disorders in particular. And within this, we created what's called the Center for Interventional Genetics. And really what we wanted to do was to have a focus specifically on unmet medical needs and working in maybe some more novel therapeutic development to try to treat these very rare genetic conditions. And this kind of started to formulate an ideas around specific genetic disorders that kind of group together in either where the gene is located or the effect of what that gene may have. So we have programs on imprinting disorders, things like maybe you've heard about like Angelman syndrome. We also have programs in where maybe there's only half of the amount of protein or the gene that's being expressed. We call these haploinsufficiencies. We have programs developing therapeutics for haploinsufficiencies. And then for several years, we've been really interested in what we refer to as like X-linked intellectual disabilities. We have people in our labs that are highly interested in X chromosome biology and all of the cool things that happen during normal development with specifically in females that have two X chromosomes as one becomes inactivated to kind of account for dosage compensation. So they have the same amount of genes that are being expressed from a female cell as would be being expressed from a male cell that only has one X chromosome. And so this very interesting biological condition of X chromosome inactivation starts to present very interesting therapeutic avenues. And so my lab actually got started on this with a different gene on the X chromosome called CDKL5. And so we started working with the Lulu Foundation in 2017, understanding, what happens, how can you reactivate a gene, or is it possible to turn a gene from the inactive X chromosome back on? And we published a paper on this. This is one of the first times that this has ever been demonstrated that you could actually turn a gene back on in a very targeted way using what we call epigenetic editing. And epigenetics are the simple way to kind of think about this is you have your DNA, and then the epigenetics are all of the things that sit on top of the DNA. So things that turn genes on or turn genes off. We have the same DNA in a lung cell that we do in a heart cell that we do in a brain cell. But the epigenetics are what tell those cells which genes to have on. So that in your heart, the heart genes are on and the brain genes are off. And in your brain, the brain genes are on and the heart genes are off. And so this is how our body kind of naturally regulates which genes are active or silent. And so we can leverage this technology and take a page from biology to go and either turn genes on or turn genes off. And so really a lot of our work initially with CDKL5 was kind of on the basic understanding the mechanisms of can this be achieved, can this be done, can you actually turn a gene back on if it's on this inactive X chromosome, because the dogma for decades have been that this is a completely inaccessible chromosome, you can't get anything to it, you can't turn it back on. And the only ways that you could potentially turn it back on were with really harsh chemotherapeutic drugs that turn on the entire X chromosome. Which you could imagine have some potential drawbacks. And so once we've kind of figured out this tool of how we can do this in cells, how we can turn a gene back on, we started thinking about how we could do this as a treatment. Could this be a therapeutic? And so we started working through the different iterations of how do we get this into a brain? How do we get this into model systems like mice to show that this is capable of being done? And so we worked extensively with Jill's lab in testing this in mice and understanding what mice with CDKL5 look like. We were able to leverage our work with this into a large grant from our state funding agency, the California Institute of Regenerative Medicine, and then starting to apply this technology in a broader sense across other genes and other X-linked intellectual disabilities. And so we also have a program in MECP2 or Rett syndrome, where we're doing a very similar process of being able to turn back on that gene. And then again, what we want to do is apply everything that we've learned and all the tools and technology that we now have that we've spent years developing into CASK. And so we've done preliminary studies, we've looked at the gene, we understand a lot of the context around the gene, and we think that this is a great candidate to be able to reactivate CASK from the inactive X chromosome. And that, again, provides a lot of advantages. When we think about the X chromosome and genes on the X chromosome, overexpression is always a concern. Having the gene being transcribed or made into RNA and made into the correct protein, and having all the different types of proteins being made is always a tricky thing when we think about gene replacement or gene therapy and gene replacement, as Laura explained earlier. And so one of the really kind of the unique things about epigenetic editing or gene reactivation, in which we're proposing, is that it's your body's own natural gene existing in its own natural context that will be regulated by all of its own natural factors. And so we can more think of it as that we're unlocking this gene and allowing it to then be transcribed to create the necessary protein that's needed to hopefully make an impact in CASK. And so I think the Cure CASK campaign, there's been certain things that have been discussed or sent out to the community about what we're planning to do. We kind of develop these in two parallel tracks, in that we do it with patient-derived cells, and so we can make iPSCs, or induced pluripotent stem cells, that from patients that we can then turn into the brain cells, either brain cells that grow in 2-dimension or brain cells that grow into 3-dimensional shapes. And then we develop and we understand how we reactivate the human genome. Unfortunately, the human DNA is a little bit different than mouse DNA. And so then we develop a parallel track where we can study then how does gene reactivation look in a mouse. Because a cell is only going to tell us so much. It's not going to have motor or cognitive deficits. We can't measure those in a cell, but we can measure those in a mouse. And that's really where Dr. Silverman's expertise comes in, is understanding translationally how can you characterize a mouse for things like sleep and seizures and EEG, cognitive deficits, and then we can apply our therapeutic into the mouse brain and see if there's reactivation. And then these two tracks converge together and hopefully go down a translational track towards what we would hope would be an investigational new drug to potentially do treatments for CASK. And so really what our proposal is doing is launching this project to push us down the road to show that reactivation of CASK could have therapeutic benefit. Was that a good enough overview? Did I miss anything, Laura, that you want me to cover? I think you might be muted. Sorry.
Yeah, sorry. I thought, yeah, no, I think you kind of covered the general aspects then. I mean, we could go on to the questions now. Yeah. I don't know.
If I could add like two sort of summary points to the very outstanding explanation that Kyle gave, that helped me make sense of it in my mind, and when I'm explaining it to families a lot, like, why do you need a mouse if you are already working with human cells? And it's that cells don't behave, right? And cells don't have the symptomology that all of the affected individuals have. And so we need a living system that actually behaves and has similar characteristics that we can put a therapeutic in and then see if it, reverses it or rescues it or see improvements in it. So that's why we need the mouse and the cell, because the cells are human and we have the human sequence and the mouse has the mouse sequence. So we really do need both. The majority of strategies that are employed by most labs in, at least in the United States, is that they will have a focus on human iPSCs or a mouse cell, or, mouse neuron cultures, or just another type of cell. It's very unique that you have a mouse lab with all of the in vivo capabilities and a huge stem cell core, like with all the stem cell capabilities that Kyle has of iPSCs.
Yeah, thanks, Jill. Yeah, so yeah, just to kind of clarify, these two labs are kind of working in parallel. Is that how you
Yes.
say it, Kyle? Yes.
Yeah, it's not, and I think a lot, and Dave unfortunately couldn't join us today. He was actually, I think, he just got back from a long trip. He was actually in Australia visiting one of the CASK families there to have a discussion as he was, and so he's catching up on a lot of I think the administrative things that he does. But yeah, really we have in essence, three labs that are all have complementary expertise, complementary skills, overlapping techniques, and definitely passion to be able to advance these therapeutics forward. So really the funding is not for an individual, but for three labs that employ lots of people that will become the leaders in CASK therapeutics.
Okay, thank you. So so before the webinar, I did ask people to submit some questions. So if there's time at the end, people will be able to just ask like general open questions. So I can see there's some people with their hands up, but I'm just gonna obviously anybody can type into the somebody's saying did the recording and hope everybody's still here. Yeah, if you have a question, like type it into the chat at the top, and then we can kind of go through that at the end. I'm going to read the questions that were given to us in the last few weeks. Okay, so the first one was, what would be the delivery method? So I guess if this kind of worked, how would you deliver it into either the mouse or the human? And how do you target the healthy silent chromosomes? How do you account for skewing?
Yeah, so really the first question is the way that we've developed a lot of these tools so far in the lab rely still on AAV gene therapy for delivery. This is this is the gold standard of getting access to the brain, being able to have things cross the blood-brain barrier. And so we've developed tools in which we use evolve or new AAV serotypes or capsids that have the ability to be delivered less invasively, so into the cerebral spinal fluid or into the bloodstream via IV, and still get to the brain and have its effect. And so this is, in its current iteration, a gene therapy that still uses AAV, but you could think of it as kind of the second or third generation of gene therapy, because the cargo in which we're delivering are these epigenetic editors rather than trying to replace the gene itself. We do have projects on kind of next generation delivery modalities that are being developed kind of in the background of these ongoing projects. Should one of those become better than AAV, or should a new technology become available, really it's the cargo right now that we think is the most valuable part, the thing that can actually reactivate the gene. So in its current iteration, this would be a gene therapy, although we are working on alternatives to that. There is the majority of the time in which we think about these X-linked intellectual disabilities is that the genes that we're going to target, or the genetic variants that we'll look at, are going to be the ones that result in either a loss of function or a null of expression. And so anything that results in a gain of function would be more challenging to target. It's very difficult to target one allele over the other, so the healthy allele versus the variant allele. If the variant allele, or the gene that has a mutation in it, doesn't produce protein, we don't have to worry about turning that off. And just all of the cells we're able to turn on. And so then every cell that receives the treatment would always have a good copy on, whether or not it had a good copy to start, or if it's a cell that has the gene reactivated. This also kind of helps account for the skewing. And so our delivery approaches ideally will hit a large number of neurons. And so if it's skewed, 60-40%, where 60% of the cells have the variant on the active allele, that these cells would receive the treatment and be able to turn the gene back on. And I think, does that answer all of the questions? So ideally, we don't have to go in and try to silent the bad copy, because the bad copy is having a loss of function rather than a gain of function.
Yeah, thank you. So yeah, you answered the next few questions. So you don't need to turn off the unhealthy genes. Overexpression isn't a concern because would it be a concern? Is overexpression?
Yeah. So in all of our work in studying the X chromosome, because again, what our approach does is it more allows for the natural gene that exists within your chromosome to be regulated in its normal way. And so we do know that there's genes that will change their expression depending on the context or depending on the situation, right? But maybe you need more CASK in certain situations or at certain times of development. And maybe there's times where you need less of the CASK around. And your body's very good at regulating gene expression as we age, as we develop. And so really what we kind of think of is what epigenetic editing does is it unlocks that gene. And in unlocking that gene, then your body will take over the regulation of that gene and be able to regulate its expression. So we always check for overexpression. We always check to see if there's toxicity due to our approaches. But typically, no, we don't see overexpression using epigenetic editing.
Okay, thank you. The next question was, would the iPSCs, and I'm presuming the mice as well, would they be open science? So would they be able to be available to others once you have completed everything?
Yeah, even before we complete everything, we like to share our resources. And so we've done this in other indications in the past. And so we try to share them as openly as possible as soon as they're generated. If other labs are interested in studying the cells, or if we have to create new mice, then we would share those mice as soon as there's enough of them existing. With the cells, it's a little bit easier because we can grow and bank them. And we've done this in a number of ways. And so sometimes we'll create the cells and we'll just grow a lot of them. And then anybody that's interested sends us an email. We process a material transfer agreement and we ship the cells. And we've done this around the world with iPSCs derived from patients. We also have a really great relationship with the Simons Foundation, as well as Coriell, that have large cell repositories. And so if it's a resource that potentially a lot of people want, or if the foundation wishes that there's additional oversight of who's getting what and when and where, we can deposit the cells with the Simons Foundation, and then it just becomes an open iPSC repository, so they don't have to necessarily come to UC Davis. And we've done that with cells. We've done that with mouse models where we'll deposit them at the Jackson labs. When then the Jackson lab will maintain the mice and be able to distribute them through their own channels, just because unfortunately sometimes our administrative offices that process the MTAs get bogged down with paperwork, and so sometimes it takes a little bit of time to process those. But yeah, we would plan on sharing everything as soon as we have materials available.
Okay, thank you. If successful,
you could even add a sentence in the agreement that, if it makes you feel better, to like confirm that, and that there wouldn't have to be any delays. Like some people they'll wait to share it until they have secured additional grant funding or a high impact publication. We just share it right away.
Okay, thank you. If successful, will you go from research to clinical trial, and have you done clinical trials before?
Yeah, so the goal is to get it as close as possible and to get this to a clinical trial, to submit an investigational new drug with our regulatory agencies, as well as work with the foundations with whatever regulatory agencies may be around. We have a lot of internal expertise within the Institute of Regenerative Cures to do clinical trials and to do clinical trial manufacturing. We actually have one of the largest academic good manufacturing practice facilities. This is a facility that actually can produce cell and gene therapy at the quality and at the volume that directly goes to patients. We're currently running about 53 clinical trials with cell and gene therapy that are administered statewide here in California. We also have a great team of people that are on the other side of the wall from me here. That's part of what we call the Alpha Clinic. And this Alpha Clinic network has the ability to access clinicians and labs and industry partners across our state and across worldwide, that we have people come in to actually set up and run the clinical trials to help consent the patients and do those things. One of the things that we would want to do as the research is advancing, and as we think that there's feasibility and efficacy to be able to advance this, our labs are pretty well versed in making contact with the FDA to go through the steps of an interact meeting, a pre-IND meeting, and submitting INDs. And so we have that internal ability to do it here, but we would want to work with the foundation, identify clinicians, identify potential sites. And I don't want to skip too far ahead, but one of the things that we always think is really important to do is kind of what we call clinical trial readiness. So if there's natural histories, if there's registries, talking to the patients about what are meaningful clinical outcomes. Of course, the first clinical trials are always focused on safety. But again, you get secondary endpoints to say what's meaningful. You know, what's going to be the most meaningful thing? Is it sleep? Is it reduction of seizure? Is it language? And then making sure that we cater, you know, whatever a treatment would be that, or whatever a clinical trial would be that we actually look at those things that would be meaningful for the patient population.
Right.
Thank you. Yeah, that was kind of the next question, actually. What kind of things could we be doing in the meantime? But sorry, Jill.
Oh, I was just gonna say that I actually we have been I have tested compounds that are in clinical trial now. They just decided to use a clinician that was more local, more East Coast localized. So yes, we have gone through the process. And then you were going to ask about, so things that you could be doing now, one thing that Kyle was saying is data that would be collected through a natural history study or through the consensus of the community would be what are the most desired outcomes that you would, hopefully we can discover something and it'll be a one-stop shop and it'll treat everything, right? Everything to the fullest extent. But, if we are being, if we're going to tamper our expectations a little bit and, say, well, we think we have a really good chance of this one domain, this one particular behavioral domain or pathology being reversed or fixed or alleviated, what would that be? What do most parents find the most challenging aspect of CASK? Is it the lack of speech? Is it sleep? Is it ambulatoriness? Is it feeding or seizures or a variety of things? And so we would want to know in order, maybe what are your top 5 to 10? Does that make sense?
Yeah, thank you. Yeah, great answer. Next question. Given that you recently received a large grant to work on CDKL5, how would this affect the timeline for CASK? Although we have to fund it first. How could the CASK research piggyback on the CDKL5 work?
Yeah. And so I think in the introduction, I touched briefly on this. And so everything that we learn working in other disorders, everything that we're advancing only informs us on how to accelerate CASK further.
Right.
And so a lot of what we initially did with things like CDKL5 was learning what tools we needed to do gene reactivation. We needed to know how to deliver. We needed to know cargo. We needed, what cells, what outcomes, all of those different kinds of things. Now that we have that established, and it's not always a one-to-one correlation, but now that all the things that we've learned in the process of X reactivation with CDKL5 or MECP2, we can take those findings, we can take what we've learned and apply them directly to CASK. And so we can skip ahead of a lot of the steps where things didn't necessarily work, right? Certain constructs that we're like, hey, this is going to be better, version 2 is going to be better than version 3 or version 1. We don't necessarily have to do all of that optimization now. We know which constructs tend to work the best. How do we best deliver them in vivo, those kinds of things, or into animal models? How do we best measure efficacy in a cell, that kind of thing. So I think it only helps accelerate what we would do with CASK.
However, we can't completely leverage the personnel, the people in our lab that are working on CDKL5 or SYNGAP will remain working on CDKL5 and SYNGAP, and we will have a CASK dedicated team in each of our labs. So that is where a lot of the cost comes from, because we have to have dedicated personnel.
Yeah, okay, so we're quite lucky really that the other diseases have kind of gathered the funds and you're able to start working on that, and we literally can piggyback on on kind of their effort and kind of benefit from all the efforts that they've made in the past. So
Yes.
yeah, quite grateful. We should
It's linked. I mean, Kyle and I have a really good start on Head Start. Well, Kyle even more so than me, but we have a good head start in X-linked disorders.
Okay,
and
right.
a large group of very bright people that are really interested in them.
Okay, so that is, oh, actually, sorry, last question from, these are all from Hitomi, actually, who is a CASK mum. Are you taking X reactivation to clinical trial for MECP2 or CDKL5? It says, since you've been successful in mice, I wasn't sure how far you'd actually got with MECP2 yet.
Yeah, so we're still in the process of demonstrating, you know, the really the key efficacy and safety that would be needed to hold those regulatory conversations. And so we have constructs in both CDKL5 and MECP2 that we show that reactivate the genes in a very meaningful way. We show that we can do it, you know, in mouse cells. We're not looking at making sure that, you know, what Jill said, you know, we not only want to be able to molecularly show that we can reactivate the gene, but when we reactivate the gene, does it make one of the things that we think would be clinically meaningful? Does it improve that? And that's kind of what where we are in the process with those.
Okay. Would there be, so this is another question. Would there be a question, would there be an issue with creating reliable cell lines for CASK?
So we've worked with extremely challenging genetic conditions in the past, both X-linked and non-X-linked genes that are critically important for cell health and neurodevelopment. And we've been able to generate iPSCs and neurons and organoids from these in our lab before in the past.
Okay, so we don't
foresee problems.
Will you be applying for grants to help with the cost?
Absolutely. Yes. There's never in any scenario where we would expect foundations, families, communities to fund everything that is necessary and or involved in a clinical trial.
A thing to add. Sorry, my voice is going out. A thing to add is, again, through the CIRM network and the grant that we have for CDKL5, is once you're able to kind of insert into the pipeline, we actually have funding mechanisms to go from translational and all the way through funding clinical trials. Yeah. So we would try to kind of insert ourselves into that pipeline.
Yeah.
Okay. Do you need the entire amount upfront, which, as we know, is over $1 million, before studies can begin, or can we begin incrementally? Yeah,
so there's certain, as Jill was describing earlier, there's certain components that are probably kind of the startup costs, and those tend to be around hiring the necessary people. We've identified people to work on these projects, but just paying their salary or ensuring that they would have you know, their salary. There's other startup costs associated with importing cells or creating cells, importing mice. So there are startup costs, but then it can be paid incrementally as milestones are met within the research.
Yeah, and we've worked with, so the foundations have been working with UC Davis to make sure there are these kind of different steps that we have to get to, and they have actually broken it down into kind of smaller pots, slightly more manageable pots. So I believe the first amount of money we have to get to initiate it is something like $650,000 US dollars. I think it was around about that. That's what we're working towards anyway, as the 3 foundations. So and that will
Yeah.
enable initiation. Does that sound right?
I think it's, yeah. So I think what we had gone through was the startup costs are about $250,000, so about 250,000, and that's just to hire and bring people in, and then by the end of the first year, coming up with about another $300,000 to $400,000 to kind of keep the project moving forward.
Yeah, because obviously we don't want to start with, you know, getting like 100, 200,000 and then it having to halt before it even kind of takes off. So.
And another key with that is, and it's kind of like it's an academic thing it's kind of difficult to explain to parent communities, is that each time we bring on someone that, you know, we want to bring on someone that can dedicate, you know, 90 or more % of their time specifically to CASK, your project, and not, you know, just hop back and forth between the different projects in the lab. We have to bring them on, but we have to commit to them that they're going to have a job for, you know, at least 2 years or more, because they're going to be moving to Sacramento. They're going to be, you know, starting to live here and things. And so a lot of times we previously, like a decade ago, when we were getting started in this field, we would ask for less money or make less of a commitment to those individuals. But then we found out that we had high turnover, which is never good for anybody in any industry, business, research, especially not research, because then our time in training them and bringing them up to a level of expertise was wasted. So we were wasting time and we were wasting money. And so that's something we have learned to avoid without making it impossible.
Yeah, thank you. Yeah, we appreciate that. Okay, right, we're coming to the end of the questions. So one mum says, well, we kind of know the answer to this. Is there any new research? She's got a son. Is there any new research for treatment for males? She said, is this research only for females? And do you know of any research for treatment for males?
Yeah, so at the time, this project is going to be focused on females, and so understanding, you know, again, anytime that we think about reintroducing a gene, starting with the females, it's just going to be, we're going to have a better understanding of what happens with gene reintroduction, what it can do to rescue some of the phenotypes, how the cell architecture looks. And then I think once we know a little bit about, you know, the necessary levels of reactivation that are required, that then this research can be expanded to males. So kind of in our phase 1, we're going to focus on females and X reactivation. As we learn more about that, we have an amazing gene therapy center here that I'm a part of. Once we understand what's necessary for rescue, then applying that knowledge to the male condition will be more straightforward. So starting with females, but yeah, we understand that there's boys affected too, and we haven't forgot about them.
Yeah. And as far as the in vivo model goes, every litter that a mouse has, there's males and females, and we don't just preferentially like toss anybody. So if we have them, we're going to use them.
Hi, I have a question, everybody. Before
Sure.
you end questions, thank you for what you're doing. My son, Anthony, is a great kid. And I wanted to know if you guys are going to be building on or working with Dr. Konark Mukherjee at Virginia Tech. He's been doing a lot of science research into the CASK genes. And then there's a doctor's name that escapes me right now over in Baylor College in Texas. Or are you starting at something completely different? And then also, I mean, my input is that you need to study males and females at the same time, because in my opinion, to save one life is to save the world. So if you save my son's life, you save the whole world.
Yeah.
Yeah.
So I think to answer the first part with understanding or collaborating, is that we always collaborate. We have extensive collaborations with labs all over the place. We've met with Konark at Virginia Tech before, I think a lot of the work that he's establishing will be critically important for us. And so understanding what happens, what goes wrong, what are the targets, what are the functional outputs that happens on like a molecular biology level only informs us or helps advance our research more if we're able to reintroduce the gene, and then we're going to be able to see those pathways or those targets or those substrates be rescued. And so that level of work is great. I don't know who it is at Baylor. We have a lot of really, really great colleagues that we work with at Baylor. You know,
like Ann Anderson at Baylor is like one of the leading pediatric neurologists in the country, and she's like in both of our phones. Like we could definitely get in touch with the right people at Baylor.
Yeah, and I serve on some committees with Huda Zogby and Rodney Samako. We work with on mouse models and in other disorders. So, and they're all at Baylor. So we know that community well. And of course, you know, as we create resources or tools, we would want to provide those to anyone else. I think what our lab specifically does that's maybe a little bit unique from some of those is, you know, we try to draw a little bit more of a straight line of what would be a therapeutic, right? And what would be a genetic target for a therapeutic. And to your point, as we're starting to develop our therapeutic platforms and our treatment modalities, as they become relevant to both sexes, I mean, we're not going to exclude anybody, like Jill said. We'll try to advance everything as quickly and as efficiently as we can.
Okay. Well, we appreciate that. If there's anything we can do to help, we'd like to be involved as well. I want to know, my son has macrocephaly, but he has a CASK mutation. It's the only clue that we have about what's going on. They've done whole genome. They've done every other available test. And they've tested him for everything. The doctors think he might also have leukodystrophy, but there's no genetic change there that they found of any of the leukodystrophies. The only clue we have is CASK. So have you come across CASK with macrocephaly at all, or CASK with any other diseases, or where it looks like possibly other diseases?
Not so far in our work, no.
Yeah, not humans, but we will be, once we have the mice and the project set up, we will be in vivo doing MRI scans on them in both males and females. So
thank you very much. I appreciate it.
You're very welcome.
So I've just got one last question, and then we'll quickly go through the chats. So the last question, oh, sorry, I'll just make myself back. So very last question, I think probably on lots of people's minds is, should a genetic therapy treatment be identified? Well, basically, if it would work, what's the oldest approximate age you would expect the treatment to produce some benefits to those with a CASK2 mutation?
Yeah, and this is such an outstanding and tricky question to answer, and one that I think we'll learn more about once we kind of launch the project. And so in other indications where it's not necessarily seen as degenerative, right? As long as the cells are not going away, the brain is incredibly plastic and has the ability to remodel and make connections throughout life. And even these critical windows are much bigger and broader than I think we even thought they were 5 years ago. Depending on, you know, how things go, how a clinical trial would be designed and developed, you know, we would try to include and have the inclusion criteria for who could receive the treatment be as broad as possible. To answer, you know, what does restoration of CASK look like? These are studies that we'll do with Jill with the animal models, right? And so we'll be able to assess what happens as the mice age and we introduce a treatment. Do we have to introduce the treatment very young? Or what happens if we introduce the treatment when the mice are much older? And then also within that realm is, you know, what can be rescued late versus what can be rescued early. So it's such an incredibly tricky question to address and something that we don't know. But something that we'll study.
For every disorder. obviously, you know, there's that an overlying logical theory that the earlier is better. Like, it's better to go earlier. But we've both seen multiple examples of adult animals showing rescue when they're adults, and so in that way, it makes me more optimistic that not everybody has to be you know, diagnosed and receive treatment within like a year. Because most people don't even get a diagnosis until like, you know, 5 or 6. It takes a long time for people to figure out that they need to get complex genetic testing and.
Right, so I think if we head to the chats, can you see the chats, Kyle?
Yes.
There's a lot, so you have to I'm scrolling down to the bits that aren't explaining how to get onto Teams. I think it's Alexis first.
Yeah. So do we plan on testing in larger animals? Yes. And I think one of the things that's really unique with epigenetic therapies or epigenetic editing is you don't necessarily need a large a disease model in a large animal, right? So we don't necessarily have to make, you know, like a pig or a primate that has a CASK mutation, because all that we're really trying to assess is what happens at the epigenetic level. Can you turn back on a gene? We have one of the 7 national primate research centers that we're actively working on these projects with. And so we'd be able to test, you know, delivery, safety, and what we call target engagement. Does our therapeutic do in a large brain what it does in a mouse brain? So yeah, we have the ability to translate that directly through large animals, and that would be something that you know, we don't have in the funding from the foundation, but something that we would do in the clinical trial development phase. Okay. Yeah, and then with regression and being degenerative, so the regression phenotype is something that we see in other X-linked intellectual disabilities. Rett syndrome is kind of the classic for this, and this would be something that we would be able to study with Jill and in the mice. Also, the case of it being degenerative, you know, again, as we're pushing forward as a therapeutic development, we'll be doing things that looking at like brain volume and imaging. And looking to see whether or not we're seeing, you know, loss of cells and or rescue of cells. Okay. Do you just want me to go down the list, Laura, and kind of read them out loud and then answer?
Yeah, I'd help you. I can't see them.
Yeah. If they're Jill questions, I'll say, Jill, this one's for you.
Okay.
Are you looking at mosaicism with these therapies too? Yes. That's one of the key features of epigenetic editing, is actually relying on the mosaicism of being able to activate the cells that have the variant or the mutation on the active allele. And we have a lot of really cool assays that we can look at mosaicism of the patterning of the brain and what cells are active and inactive, and being able to tell what are the effects of the, you know, a healthy cell next to a cell that carries the mutation or has the mutation active. Estimated timeline to get this to a patient, or too far off to be able to say? Too far off to be able to say. And so research is never linear. We always learn something. There's always something new that comes up. We're hoping this is a 4-year project as it's written in the proposal. We hope that in those 4 years we have the necessary data to be able to go to the FDA and start to hold those regulatory meetings, getting us through like the Interact or even like the pre-IND meeting, in which then the regulatory agencies would tell us what data that they need to be able to make an accurate assessment whether or not they think this will be, if the benefits of whatever this therapeutic would be will outweigh the risks. The world of gene therapy and using epigenetic editing, things like CRISPR, to be able to accomplish these goals is relatively new. And sometimes the FDA will ask for a lot of additional information and a lot of additional experiments, and other times they won't. And so we can't necessarily plan for what the regulatory agencies will look at within this therapeutic. and what additional things that they'll ask. But we're hoping at the end of the 4-year funding that we're ready to, or we'll be holding those meetings with the FDA to discuss what the steps forward to get this to a clinical trial would be.
Okay,
I think
so Isaac's
oh yeah,
vote.
go ahead.
Yeah.
Yeah. So I think we answered those ones. We answered the aging one. I think, again, we hope that this would work in a developed brain. We hope this would work and be amenable to older patients. In other X-linked disorders, we see that reactivation of a gene rescues a lot of function even in older age. And so if this holds with a lot of the other X chromosome genes that we study, potentially the older patients would benefit as well. Mutations and microdeletions, yes. And so our approach is mutation or deletion agnostic, again, because we're only going to be targeting that intact, healthy version of CASK to turn that back on. Access new information, progress, potential treatment, participation, and therapy progress. We love interacting with families. We love sharing our data and talking about what we're doing. We love our jobs, Jill and I and Dave. We're very passionate about what we do. We love talking to families and sharing, not only the successes, but the roadblocks and how we plan to overcome them. I think once we launch
the can only take so much.
And I think, once we launch the project and we start collecting data, I think with the CASK, the 3 CASK foundations that are going to be supporting this, we can set up, recurring, check-ins and progress reports if they want, if the community would like webinars, a couple of times a year, we're happy to share that and present. And also if there's ever conferences, family conferences, or events, we love going to those and meeting everybody and discussing our work.
Ditto.
Yeah. Oh, and if there's mouse models at other universities, we would love to get in contact with them and share. I think that was one of the questions is, the more resources that are readily available that we can import, that helps us. We'll probably still create some of our own because we have some unique tricks of things that we like to do. But yeah, if there's other mouse models and information that's being learned, being connected with those individuals, so everybody can move forward quicker and more efficiently. We like to be open and collaborative as much as we can be. Thank you for your work. CASK and PCH related. I can't answer that from like a clinical perspective, whether or not we see a direct correlation between the mutations in CASK and PCH. Yeah.
Would you like me to try and answer from my non-medical background? So CASK is obviously the gene and PCH is the condition that can be seen on the MRI. So it's just, as from what I understand, it's just the kind of description of what the brain looks like, which is what the majority, I think, of our children have. So, but PCH is caused by lots of different genes. So there's like PCH type 1 caused by one gene, PCH type 2 caused by another gene, etc. And CASK is just one of the many genes that can cause PCH. So I don't know if that answers that question, but
yeah, thank you. So what do we have next? Timeline to start up clinical trials. What does it depend on? Any idea if health insurance would cover any of the gene therapy? So the timeline to start, I think as soon as the foundations say go, we're ready to start. We have people that are very enthusiastic to start working on CASK and the therapeutics for reactivation. And so, we're happy to participate. We're just kind of waiting on the foundation to say we're ready to go. And then clinical trials, that's something that we don't know. Again, if everything goes incredibly smooth and we are able to see rescue and therapeutic benefit, it's really going to come down to the regulatory agencies and what they view and what additional information that they would need to be able to make a call of whether or not they think that the benefit of this approach would be worth whatever risks, right? And I think, anytime that you're starting about early clinical trials, there's always going to be inherent risks. There's inherent risks with gene therapy. There's inherent risks with any potential novel treatment. And so it's just whatever information that a regulatory agency would say, this is worth the potential benefit, the potential outcomes that this could provide to people suffering with CASK variants would be worth potential risks of a clinical trial. Any idea if health insurance would cover any of the gene therapy? This is a very hot topic that we discuss at length with a lot of people. Gene therapies are becoming inherently more and more expensive, and they're really starting to tax a lot of healthcare systems. This is another mission that we have here at UC Davis, is making these therapeutics more accessible in terms of driving down the cost. We've done this with CAR T therapeutics for cancer, in which we're able to manufacture and administer CAR T therapeutics for about a fifth of the cost as what larger biotech companies are charging and are charging insurance from. And so with gene therapy, with what we would do with CASK, we're also working in the realm of significantly driving down the cost to make these types of therapeutics more accessible. That's something independent of CASK that would directly apply to what we're doing here. And that's just one of our missions at the Gene Therapy Center here at UC Davis. And then, of course, clinical trials are typically covered by the sponsor. So that would be us or CIRM or a biotech company that we would partner with. We would try to treat as many people under a clinical trial as possible. And then once it gets to the question of health insurance is kind of once it gets through like a phase 3 clinical trial and approved at the market, or approved for market. Okay, how is this treatment different than the possible treatment Dr. Konark has proposed? I think a lot of what Konark is looking at are more small molecules that may affect either downstream function of CASK. And so more not restoring the gene itself and all of the function of the gene, but restoring some of the pathways in which the variant affects. And anything that we think would improve the quality of life or help address some of the symptoms, we're all for. A lot of what our focus on between Dave, Jill, and myself is things that specifically target the underlying genetics and rescue the underlying genetic condition, which then we're not necessarily dealing with pathways and off targets or side effects. Okay. Is the reactivation a one-time procedure process? Yes. Epigenetics are incredibly cool, and we could spend hours talking about how much we love epigenetics as a biological tool. But epigenetics is something that we refer to as kind of being metastable. So as soon as we're able to do epigenetic edits, this would be long-lasting. Also, the way in which we would deliver this to the cell would be a one-time treatment. And so versus a lot of other types of approaches where maybe you have to get treated, multiple times a year, we're designing this to be a one and done. And so you would get treated once, and that treatment should last for the life of the patient. Did I make it through all of them?
You did. Yeah.
Okay.
Thank you so much. And I think that was a really nice question to end on. So unless, I don't think there's any more questions coming through, so it's getting quite late. Yeah, so thank you so much, Kyle. Thank you so much, Jill. I'm so sorry people couldn't meet you in person, Jill.
No worries.
You've got a nice voice, so
in the future, I hope.
Yeah. And thank you to everybody who came along. I think we had about 40 people, which was amazing. So it really shows, the support of the community. So I hope everybody's feeling really inspired. We really need your support. So one of those last questions was, when is it going to get started? It's going to get started when we get the funding, and the funding is going to come from the community and everybody and anybody. We need your help. So just to remind everybody, there's the UK charity, which I head, CASK Research Foundation. There's the American foundation called ACNRF, the Angelina CASK Neurological Research Foundation. And they also are in Australia. And we have the French Foundation, Association Enfant CASK France. Apologies, I have no French accent. However, even if you're not in any of those 4 countries, people can still donate and people can still fundraise, because we all can accept money from, like, money globally. It's just if you're in those countries, all 4 of those charities will, you can receive tax relief if you donate. So that's the benefit. But anybody can donate, anybody can fundraise, anybody can support us. So please keep in touch, find which charity kind of suits you best for your location, and we'll be in touch. So thank you, and thanks everybody for coming.
Sounds great. Thank you so much. Thank you for
Thank you,
organizing.
everyone.